Hello and welcome to this masterclass in clinical genomics and this session on focusing on metagenomics. My name is David Nestor. I am a resident physician in the clinical microbiology and a PhD student. My research is focusing on using metagenomics for diagnosis of bloodstream infections and sepsis. I will not have this presentation on my own, but together with Paula Mölling, who is a molecular biologist and associate professor, and Frans Wallin, who is a bioinformatician here at our lab. And my part of the presentation will focus on giving a brief introduction to the topic and also talk something about the potential clinical applications of metagenomics. So first and foremost, what is metagenomics? And it's quite difficult to give a short description of the topic. Looking at the mesh term for metagenomics, it says that metagenomics is the systematic study of the genomes of assemblages of organisms. Could be quite difficult to understand, but two keywords here is genomes and assemblages. We're focusing on the genetic material, the genomic material, and also we have the potential to analyze assemblages or various species at the same time. And importantly, metagenomics is not limited to any particular species. We can analyze bacteria and fungi or eukaryotes or viruses at the same time. Any organism with DNA or RNA, in fact. Some of the terminology here, we talk about targeted metagenomics, which is in fact mostly 16S or ITS sequencing for fungi, or untargeted metagenomics called shotgun. We also used NGS or next generation sequencing technology. Those of you who want some more of information about the history of sequencing, you could look at this paper. It goes through that in more detail. The advantage of next generation sequencing is the possibility to sequence different DNA fragments in parallel, in contrast to Sanger sequencing, where we only can sequence one particular amplicon or part at the same time. And the two main platforms for NGS is this nanopore technology, where DNA is transferred through a nanopore in the membrane, where you record the difference in electric current, or this sequencing by synthesis technology, mainly used by Illumina, for example. So what are the possible clinical applications of metagenomics? First, we could mention whole genome sequencing. In fact, whole genome sequencing is not really metagenomics in that sense that you only sequence one bacteria at a time, for example. But we use that in clinical routine where we for strain typing or outbreak investigations, and we use the same platforms for this purpose, although we only look at one species at a time. Another application of metagenomics could be analysis of whole microbiomes and the association with disease. This is not used in clinical routine, but there's a lot of research in this area going on, looking at, for example, gut microbiota and its connection with different diseases and so on. My main focus for this presentation will be to use metagenomics for pathogen detection directly from primary sample. And there has been a lot of expectations for using metagenomics for this purpose, as it has the possibility to detect any organism present in the sample. So what are the potential promises for using metagenomics? First, we have the possibility to have higher sensitivity with metagenomics than with, for example, culture. For example, in patients who are on antibiotic treatment, who might not have viable bacteria, or difficult to culture organisms like intracellular bacteria or viruses. One disease group here that could be mentioned is endocarditis, and recently metagenomics was proposed as a way to diagnose endocarditis because it can be caused by difficult to culture organisms and so on. For the interested, you can read more in that paper. We also have the possibility with metagenomics for genotypic prediction of resistance, in that we sequence the genome and we find resistance genes. It is in theory possible, but it's also at the same time difficult because not everything that is present in the genome is expressed phenotypically in the patient or in the infection situation. Also, if you, for example, find a plasmid, if you sequence the flora of in the intestine, for example, and you detect a resistance plasmid, it's very difficult to link that plasmid to a specific species, as it's not part of the core genome of the bacteria. Another possibility with metagenomics is to detect polymicrobial infections, and that is something that we don't know so much about. We think about it like one pathogen causes one type of disease, but in fact, maybe we have interactions between different bacteria or between bacteria and viruses, bacteria and fungi, and so on. And I think that maybe we will learn more about this polymicrobial infections by using metagenomics. That's a field of research that will be very interesting to follow. Also, as with the other molecular techniques, metagenomics can be performed very, very fast, maybe within a few hours. However, I think the biggest difficulties with this approach is to get enough logistics and automation for this to happen, so to speak. It's difficult to have the samples transferred from the emergency department to the laboratory and undergo all these analytic steps in a short time. Maybe the technique in itself does not take so much time, but the transportation will be the main difficulty. So looking at what has been published in this area, this is a paper from last year, a bibliometric analysis of all papers focusing on metagenomics for pathogen detection. And you see here a very heavy incline during the last years. It has reached a very much of research interest. And one take-home message from this paper is that the majority of papers is from China. I get the impression that metagenomics is much more used in clinical practice in China. And also we have a lot of the technical development going on there. In this paper, they also identify hot topics where metagenomics has been used. Taking them one by one, pneumonia, of course, it's a result partly because of the pandemic, where the differentiation between viral and bacterial infection have been very interesting. For tuberculosis, we know that Mycobacterium tuberculosis is very difficult to culture, and also prediction of resistance has been found very easy to be performed with metagenomics. You can identify the resistance genes, and they are clearly coupled to phenotypic resistance. For CNS infections, it's mainly because cerebrospinal fluid is considered a sterile fluid, and CNS infection can also be caused by a wide array of different organisms, both viruses, bacteria, and yeasts. So it has mainly been used for this purpose. Also for pediatric infections, I think partly because children can be infected by a wide type of organisms, and also often the sample volume might be very small from these small children, and these molecular techniques does not require so much volume to be performed. That what might be one of the advantages for this purpose. We sometimes talk about metagenomics as an unbiased approach or an hypothesis-free approach. And when you read the papers, you get the impression that it's like they've sampled the patients and then they do some kind of metagenomics. Quite often the method section is poorly described and you have not really an idea of what they have done. But in the end, you get very complicated figures and graphs of the results, which can be very difficult to interpret. And I would not say that this method is unbiased. We all know also that this metagenomic workflow is very complicated. We have many different steps with DNA extraction, library preparation, sequencing, and so on. Rather than saying it is unbiased, you should think of it as biased in many different ways, in very complicated ways that can be difficult to interpret. So in order to understand these graphs and these papers, you both need sort of a clinical interpretation of the disease and the patient, but also an interpretation of how the methodology was performed. We need to combine that in a good way to get an accurate understanding of what has been going on. Just to give you a brief some examples from the literature of the difficulties with metagenomics. This paper from last year was from China, and in this paper they used sputum samples for lower respiratory tract infections. They had both spiked samples, contrived samples, and also clinical samples. And they sent these samples to, I think it was 120 laboratories performing metagenomics, and then they gathered all the results. And looking at this figure C, you see that here is all these S1, S2, S3, S4 is the different samples. And the number here is the number of false positive microbes reported by the laboratories. So in sample 4, for example, they reported 180 false positive microbes, probably a lot of different viruses and bacteria and so on, which is very difficult to interpret in a good way. Also, looking at this heat map, you see the differences in the report of bacteria from the different labs. And the main message here is that the reports differed very much from laboratory to laboratory, which clearly says that it's very difficult to reproduce the results. And I do not think we should take these results as disencouraging. I prefer to think of it I prefer to think about them as we have very much to learn, still very much to learn of how to use these metagenomics techniques properly. And I think it's very good that these types of papers are published. One example also from my field of research for sepsis. This is a paper where they used metagenomics for sequencing of patients with sepsis in intensive care units. And the main interesting thing here is that 14% of the patients had more than 7 pathogens in blood. Usually when we do culture, we find 1 or maybe 2 pathogens and we try to interpret that, but we have no clinical experience of how to interpret 8 different pathogens at the same time. So of course, the methodology is there, and maybe that we need in a way to understand these results and take them into. How should we use them? That's the question. How should we use it for clinical purpose in the clinical situation? So my main take-home messages from this is that you should think about metagenomics as a pivotal new way of analyzing biological samples. It is different from both culture and other molecular techniques. And in order to understand them properly, you both need we need a technical understanding of how the analyses were performed and how they work before we sort of apply them to the clinical routine. And that was it for me. I will leave over to Paula.
Okay, now you have got an introduction in metagenomics and heard some about the clinical applications. So I will talk about more the technical part, the wet lab, the sequencing methodology. So my name is Paula Mölling, and I'm a molecular biologist, associate professor at Örebro University Hospital and Örebro University in Sweden. And to start with, we go for the wet lab procedure. That is really important. So here we have to take account the collection, storage, and handling of the sample. What kind of sample matrix, what kind of sample material you will be used to be able to answer the clinical questions, and the sample preparation, and also the metagenomic approach, if you will go for a target or untarget approach where you sequence everything in the sample. And this is really crucial steps to ensure a high quality and contamination-free sample as possible. So we start with the collection, storage, and handling of sample. If we go to the first step where you sample the material, you have to make sure it's as sterile as possible to be able to avoid contamination, because that will follow in the entire procedure. And the transport time, that is often in room temperature, here we have to ensure it's as short as possible to avoid as much as possible the risk of degradation. And for the storage of your sample, for short-term storage, you may store it in 4 degrees. And for a whole blood sample, for example, it's not recommended to store it for longer than 3 days. And if you're aiming for bacterial pathogens, you can store it for long-term storage down to 20 degrees. But for the RNA, you may use even lower degrees, like 80 degrees. And for longer term storage, it's also recommended to freeze it into smaller portions to avoid the cycles of freeze thawing. And for the sample matrix, this depends, of course, on the clinical questions, what kind of sample material that you will use. For example, for sepsis, bloodstream infection, or meningitis, you may use whole blood or plasma, or even cell-free DNA. But here you have to be aware of, it's not very clear the clinical relevance of the cell-free DNA. If you got a really high amount of it, of course, this increased the possibility of clinical relevance. And then you can also use, for example, the cerebrospinal fluid, so meningitis. And if you have a non-invasive infection, you can go for nasal or skin swabs. Or if you know it's a local infection, you may also need to use the tissue biopsies. And then we also have to take into account that the duration of detected DNA depends on the sample matrix, but also it can vary between the different pathogens. So here are 2 examples from a study that we have done on sepsis patients, where we have looked at the duration of Staph aureus, E. coli, and Mpox, for example. And here we found that the Staph aureus we were able to detect the Staph aureus DNA for up to 6 to 8 days, compared to E. coli and Mpox. It degrades much faster, and we could only detect it a few days after admission to the hospital. So for the sample preparation, we have to think if we need pre-extraction steps. And if we have a low amount of pathogens in the sample, for example, we need to go for a higher volume. So we may need kits that can extract from high volume. Or we can add a concentration step with centrifugation, or maybe it's just enough to purify it. And then we have also decided if we need human depletion or not. And for that, it's a variety of methods have been used, like for the DNA, saponin, and centrifugation. But here we have also be aware of, for some pathogens, this may not be optimal, for example, viruses. And then if we have, for example, hard to lyse pathogens, we may think of pre-lysis step. And this we can do mechanically or enzymatically. And here we have to be aware of, if we will use long read sequencing next step, you may not use too hard mechanically, because this will shear the DNA into smaller fragments. And for the nucleic acid extraction, of course, we have to decide if we would like to go for DNA or RNA, or maybe we need both for further analysis. And the library preparation, we can choose to do it target or untarget, where we sequence everything in the sample. And it's really important in the sample preparation that we use controls in metagenomic sequencing. So always include a positive and a negative control. At least a negative control should be used from the first step of the sampling and follow the entire protocol to the sequencing. And all those steps need to be validated for each sample matrix and application. So now we come to the metagenomic sequencing. As I mentioned before, it can be performed in two different ways. If we start with the target approach, and here we also call it amplicon sequencing. And for example, we can use the 16S gene for bacterial species identification, or the ITS, internal transcriptome segments, for fungi. And this is positive because it's often more cheap if you compare to the target approach. It doesn't require so much DNA. And since it's include an amplification step, it may also increase the sensitivity. And often there is no human DNA to take account to. But it's also limited resolution. You can only find what you are looking for. And that is often not antimicrobial resistant genes. And then for the second approach, target, but also it's called shotgun sequencing. Here we sequence all material, and that is really good because we are able to identify all bacteria, virus, fungi, everything that is in the sample. And if we are lucky, if there is a huge amount of the pathogen, we can also be able to predict some resistance and virulence, and maybe some further typing of the pathogen. But this often requires more complicated bioinformatic analysis, and we also have to deal with the contamination issue. We need more controls, and much more expertise is often needed. And we have this huge amount of human DNA to take into account. And for the sequencing methodology, for the short read sequencing, we have two different generations. The first generation, it's mostly Sanger sequencing. The second generation, we've started to mass parallel sequence the fragments, but this is often more shorter fragments. And for the third generation, that is often more and more in clinical diagnostics, it's the third generation with long read sequencing. So if we compare the short read to long read sequencing, and here I have chosen to compare it with the ONT, Oxford Nanopore Technology sequencing, since that is more advantageous to use in clinical metagenomics. So if we compare the quality, the long read sequencing gives fewer reads, but often longer reads. And the fewer reads, of course, it also gives a lower coverage. But this is really good when you have long reads. You may also be able to find both the ID, the pathogens, combined with the resistance in the same read. And the long read ONT sequencing has also higher error rates, unfortunately. But here you can be sure that you use the latest chemistry and software, because the higher rate the error rate has been improved by that. And the turnaround time, the long read sequencing is often more rapid if you compare to the short read. And the cost between the different technologies often depends on the throughput and application. So for short read sequencing, you often need much more samples to get this cost effective as possible. While in the ONT sequencing, you can sequence only a few samples at a time. And the choice of sequencing platform, of course, affects the depth, accuracy, and also the speed of sequencing. For processing the sequencing data, there's a lot of steps in the data processing, like the quality control, alignment, sample. You have to put all the small reads together, the annotation, identify combination, and for this, it's really optimal if you could set some kind of threshold. And there's a lot of bioinformatic free tools out there and softwares, but for metagenomics, you really need robust bioinformatic tools to process the huge amount of data generated in metagenomics. And all those steps you will hear more about in the next session. And there's also a challenge to handle all data. So it's really preferable if you can use joint solutions for IT infrastructures and pipelines. This can be, because this can be a real problem for the smaller laboratories, for example, that has not developed the IT infrastructure locally. So those joint solutions can be really good for them. And for the data interpretation, it's really important to be able to distinguish the clinical relevant pathogens. And for this, you need to define the background noise. And here we can talk about the lab biome as well as the kit biome. And the lab biome can be if you sequence a lot of specific pathogens at the laboratory level. You may talk about the lab biome that can also contaminate metagenomics. And if you have some contamination in the kit, in the extraction, or in the library preparation, here we can also talk about kit biome that is related to specific kits. And the understanding the microbial ecology is also important. So here you have to study the microbes in the environment and how they interact with each other. And the very, very important step is to get the results to set it into the clinical context and the relevance. So here you have to compare the clinical data to be able to draw any good clinical conclusions about the pathogen that you find. And as we talked about before, the cost and turnaround time, here is really important to balance the cost of sequencing and analysis with its clinical benefits. And the turnaround times are here really crucial for the timely decision making. So here we have to develop strategies for cost-effective implementation. So now you can take the time and maybe pause this presentation and think about the pros and cons for target vs untarget metagenomic sequencing that I have talked about in this presentation. So lastly here, I will give you some take-home message. I think the clinical metagenomics really has the potential to identify unexpected microorganisms and interfere their susceptibility to antimicrobials. But for this, we may need some further optimization. So here it's really important that we may optimal all the steps, and that may have to be done for different kind of sample matrices, different kind of samples, as well as clinical questions. So by addressing these clinical aspects, clinical metagenomics can become a really powerful tool for diagnosing and managing infectious diseases. So this was all for this section, and thanks a lot for listening. So now we come to the next session, where you will hear about bioinformatic analysis.
Hi, my name is Olivia Andersson, and I work as a bioinformatician. And in this segment, we're going to focus on bioinformatics approaches within metagenomics. So bioinformatics workflows are often organized into pipelines to handle their complexity, and these pipelines automate analysis by integrating various tools and databases. Most pipelines are open source and available on platforms like GitHub, allowing users to adapt them to their specific needs. In addition, workflow management systems like Nextflow are commonly used to manage these pipelines effectively. Nextflow is a domain-specific language that simplifies the development of these complex workflows. A popular initiative within the Nextflow ecosystem is NF-Core, which is a community-driven collection of high-quality, pre-configured pipelines that follow standardized practices to ensure reliability and easy adaptation. A typical pipeline starts with pre-processing, during which quality assessment of the sequencing data is performed. One popular tool for this is FastQC, which examines various metrics to evaluate the sequencing data, including GC content distribution, sequencing lengths, and overrepresented sequences in order to identify potential issues, such as contamination or problems with sequencing adapters. One common issue with raw sequencing data is the presence of these adapter sequences or low-quality bases, especially toward the end of the reads. And in the case of 16S sequencing, primers used for amplification may also need to be removed. To handle this, tools like CutAdapt, Adapter Removal 2, and PortShop can be used, and they identify and remove adapter or primer sequences through substring searches and trim away any low-quality bases, and removing these is crucial to avoid biases. And once the data has undergone quality checks and low-quality bases or primer sequences have been removed, the presence of host DNA can be addressed. As Paula mentioned in the previous segment, human DNA can still be present in the sequence reads, although human degradation steps have been performed. And addressing host DNA is particularly crucial in metagenomics when shotgun sequencing is used, as the host genome often is more abundant than the microbial DNA, which can obscure the analysis later on. To remove host DNA bioinformatically, the sequencing reads are aligned against a reference genome, and any reads that match are flagged and removed to ensure that only microbial DNA is left for further analysis. Biotype 2 is an aligner that can be used for this, and it uses a Burrows-Wheeler transform to index the reference genome. It employs a multi-seed strategy, which involves breaking the reads into smaller segments or seeds and searches for approximate matches within the reference genome. This method increases the likelihood of alignment even in regions with mismatches, small insertions, or deletions. Minimap2 is another widely used tool for aligning long sequencing reads, and like Biotype 2, it uses the Burrows-Wheeler transform, but uses an anchored seed approach instead. It identifies long exact matching segments against the reference, and then extends the alignment around these, whilst allowing for mismatches, insertions, deletions, and larger structural variations, such as rearrangements. And after alignment, both Biotype 2 and Minimap2 outputs the mapping of the reads to the reference genome, and reads that align to the host genome are either discarded or flagged for further examination, while the remaining reads, so those that did not match the host genome, are considered microbial and can be used for the next step. For the taxonomic classification, the reads are compared to large curated reference databases of microbial genomes, such as PLUF-PS, Green Genes, and Silva, which contain sequences from a wide range of organisms. Using taxonomic classifier tools, the sequencing reads are aligned to these reference sequences, allowing for taxonomic assignment. And two popular tools for this is Kraken-2 and METAFLAN. So Kraken-2 uses a k-mer-based approach, dividing reads into fixed lengths of substrings or k-mers that are mapped to the reference database. Each k-mer is assigned to the lowest common ancestor of all genomes in the database containing that given k-mer. And this enables Kraken-2 to classify reads to the most specific taxonomic level. To improve accuracy, Kraken-2 also employs space seeds, which allows for gaps between matching k-mer positions. And this enhances sensitivity in highly variable genomic regions, making the tool robust for complex microbial classifications. METAFLAN uses another approach by relying on marker genes that are unique to a specific taxa, rather than using entire genome sequences. Instead of mapping k-mers to a reference database like Kraken-2, METAFLAN focuses on identifying a predefined set of conserved taxonomic marker genes that are highly specific to a certain taxa. And these marker genes used by METAFLAN are typically selected for their high degree of conservation and their relative uniqueness to that taxon. And once the reads have been aligned to the reference databases, they are assigned their taxonomic labels based on their best match. And in addition to taxonomic assignment, the relative abundance of each identified taxon can be estimated by counting the number of reads assigned to each taxonomic group. When Kraken-2 is used for taxonomic assignment, Bracken can be used to help refine these estimates by accounting for sequencing depth and potential sequencing errors and biases that might affect the quantification of taxa. Bracken improves upon the basic read counting methods by using probabilistic models to adjust for the influence of these factors, leading to a more accurate abundance estimation, especially in datasets that have uneven read distribution or low sequencing coverage. And as stated earlier, the complexity of bioinformatics workflow often necessitates the use of pipelines to streamline and automate this multi-step process. And as metagenomic workflows can vary significantly depending on the research question or sequencing approach, different pipelines are designed to address these specific tasks or data types. For example, shotgun metagenomics generates highly complex datasets that require comprehensive tools, like those provided in the NF-core tax profiler pipeline, which accommodates for both long and short reads for robust taxonomic profiling. In contrast, 16S workflows, which focuses on targeted regions of microbial genomes, benefit from pipelines like GMS16S. And when identifying antibiotic resistance genes, specialized pipelines such as NF-core FuncScan and Epi2Me Labs VF metagenomics can be used. And these pipelines are specifically suited for functional profiling. For the visualization of the results, one popular tool is Krona, which provides dynamic multi-layered pie charts for each sample in the dataset. These charts are interactive, which enables users to zoom into specific details within the sample, making them ideal for datasets with a hierarchical structure. And the layered visualization of Krona charts provides us with a clear and interactive way to interpret taxonomic diversity and distribution, helping us to identify dominant taxa, detect unusual microbial patterns, or pinpoint pathogenic microbes. And these visualizations are browser compatible, making them easy to share and interact with. And since they are so easy to use, as well as interpret and provide a per sample view, they are well suited for clinical applications. And while Krona excels at this type of interactive visualization, further analysis, and a more in-depth exploration of the result is often required and then requires additional tools. And for this, we can use softwares like R along with specialized packages like FileSec, which provides extensive capabilities for data exploration and presentation. And these tools allow for the generation of charts and heat maps to identify trends, patterns, and relationships within the complex data sets. For example, principal coordinate analysis, or PCOA plots, can be used to visualize the similarity or dissimilarity between sample groups based on distance matrices, helping us to reveal patterns or clusters in the data. Additionally, alpha diversity can be used to offer insights into the richness and evenness of microbial communities within individual samples. Beta diversity, which often is visualized through PCOA or NMDS plots, enables the comparison of microbial diversity between different samples or groups. And here you can see an example of a study that examined the microbe of infants from 3 to 12 months of age. And the alpha diversity is visualized using the Shannon index to represent diversity by combining richness and evenness, and the SHA-1 index is used to estimate the species richness. And both these metrics show significant increase over time, highlighting a change in microbial composition during this period. The beta diversity can also be visualized using a PCOA plot with the Bray-Curtis dissimilarity index. And each dot or square represents an individual sample, and the plot organizes these samples in a multidimensional space based on their microbial profiles, with closer points indicating more similar communities and farther points indicating greater dissimilarity. In this plot, the clusters correspond to the two age groups are clearly separating, highlighting distinct differences in the microbe composition across the age groups. And the ellipses, which represent the 95% confidence intervals for each group, show limited overlap, further supporting the observation that microbial communities within these two age groups differ. And ultimately, the choice of visualization technique depend on the specific question, the type of data, and its application, whether it's in research or clinical settings. And just to quickly summarize, these bioinformatics workflows are rather complex and multifaceted and involve multiple stages that collectively enable the analysis of microbial communities. However, it's not just the individual steps that matter, but how they are integrated and how the right tools are selected to address the specific research or clinical question. Numerous tools as well as complete pipelines are available for this, and given the varying differences of these tools, selecting the most appropriate one is an important factor. Beyond enabling the classification of microbial data, these tools and pipelines are integral to deriving meaningful insights. Visualization tools, including Corona and R-based packages, play a critical role in interpreting the results, and they allow users to explore taxonomic diversity, compare microbial communities, and identify meaningful patterns. And these visualizations not only make the complex data accessible, but also enables the identification of specific pathogens while providing a broader understanding of microbial compositions, adding valuable context for both research and clinical applications.